Hi guys, this is Michelni. Along with Puvi, we'll be presenting the Assignment 8 Clinical Chemistry Question 2. How is this disorder investigated and diagnosed? Explain in detail the method for sampling and testing. This disorder can be investigated and diagnosed using the tests below. Some of the tests are U3 blood test, genetic testing, fluorometric analysis, and tandem mass spectrometry. The first test is the U3 blood test, which is also known as the U3 bacterial inhibition assay. In this test, the baby's heel will be pricked with a needle to obtain a spot of blood over the specially treated diagnostic card. After collecting the blood, the sample is incubated with a genus of bacteria known as Bacillus subtilis, along with a chemical known as B2-phenylalanine. This chemical tries to inhibit the growth of the bacteria by preventing them from feeding off the phenylalanine present in the sample. If the bacteria cannot grow, the result is negative. If the bacteria can grow in the sample, the result is positive, which indicates the presence of phenylketonuria in the baby. This test is an inexpensive, simple and easy to use test that is applied routinely. Other than U3 blood tests, genetic testing also can be done. Genetic testing, which is also known as molecular testing, will be done to look at the mutations in the genes that cause phenylketonuria. This test will also look at the phenylalanine hydroxylase deficiency via the activity and functioning of the phenylalanine hydroxylase gene on the 12th chromosome. There are between 1 and 15 faults or mutations which can predict nearly 30 to 50% of genetic defects in the phenylalanine hydroxylase gene. The target identification of this test is to look at these faults specifically to identify phenylketonuria. The next test that can be done is a fluorometric analysis. This test requires machinery that tests the blood of the baby to identify the raised levels of phenylalanine. Blood sample will be collected into a heparinized tube and it will be then spotted directly onto filter paper from blood collection tubing for analysis. The blood measurements will be performed in an automated reader to identify the raised levels of phenylalanine. This test shows more accurate results than the bacterial inhibition assay. Tandem mass spectrometry is another test that has similar reliability as fluorometric analysis, but it finds the level of tyrosine. In amino acid catabolism, phenylalanine is ultimately broken down into tyrosine with the help of phenylalanine hydroxylase. In phenylketonuria, there is a deficiency of phenylalanine hydroxylase, so phenylalanine will not be converted to tyrosine. So if the value of tyrosine is lower than its normal value, it indicates phenylketonuria. This test is considered accurate as it shows the actual picture of the condition. This picture shows a butterfly needle is being used to draw the blood from the kid. This is because once a child starts standing or walking, they develop thicker skin and calluses, so the heel is no longer an option to draw the blood from. Babies and children have smaller veins than adults. A butterfly needle makes the procedure for drawing blood more manageable because the needle is thinner to accommodate for the smaller vein. Figure 1 shows how to hold the infant's hand where the grasp is as shown. This achieves both immobilization and tourniquet. Figure 2 shows the angle of insertion of the cannula, which is at the 10 to 15 degrees. Figure 3 shows the collection of blood samples where the blood is let to drip passively into the collection bottles. And figure 4 shows the aspirating blood from the hub of the cannula using a blunt drawing up needle and syringe. The sites for blood sample collection are the dorsum of the non-dominant hand, which is preferred usually, and the vein running between the 4th and 5th metacarpals is most frequently used. Figure 5 shows the blood collection tubes for babies and kids. These are our references. Thank you.